Good afternoon, everyone. Very happy to be here with you today. So, before we start with today's lecture, um I've actually uploaded the course materials for today and from the 10th of the month, and you can see the links into the chat. So, in case you want to download them, you want to check them out, you want to run the examples that we've either seen last week or the ones that we're going to check out today, feel free to do so. Um, if anyone can actually click on the links and verify that they work, this would also be very, very nice so that if they don't, I can actually find a workaround for it. So without further ado, I'm also going to start sharing my screen today. Okay, so I'm guessing that you can also see my screen right now. Is that correct? Confirmation would be
Yes,
nice.
yes,
Yes,
Okay.
it's
yes.
correct.
Okay, great, thank you. So last time, well, over this course, over the past days, we've actually checked out many different applications of machine learning algorithms on classic tabular data. Just as a reminder, we've seen what happens in a benchmark data set using Iris. And last time, we actually went on and applied everything that we've seen through Iris on a real world biomedical data set, which was again tabular in nature. And we've actually checked out many different, let's say, traditional, so to speak, machine learning algorithms and methodologies. So today, we're actually going to check out neural networks, artificial neural networks to be exact. And we're going to check out two different applications of these on biomedical problems. One is actually going to be the starting one is going to be on biomedical images, specifically on CT scans for COVID-19, and we're going to talk about that a bit later on. The second one is going to be a neural network architecture that is used to denoise electrocardiogram signals like ECGs. So let's start with the first one, with image classification, specifically COVID-19 image classification. Just as a parenthesis here, again, this is an HTML file that I extracted from a Jupyter Notebook. So what you see here is basically the product of a Jupyter Notebook. However, because of the fact that training actually takes quite a lot of time, and by a lot of time I mean about 1.5 hours, I thought that it would be a better idea to just run it myself and go through the code and the interpretations as well as the results, together here. So, of course, in the course materials, I also have given you the original notebook in case you want to run it, but we're going to check out as well as the HTML. But today we're basically going to check out what's going on through this HTML file. And the same thing is going to happen for the other neural network architecture that we are going to use or that we have used for signal denoising. Is there a question in the chat? Oh, okay. So thank you, Andrea. So glad to hear it. So thank you very much for the confirmation. Okay, so um, the data set that we will be using today comes from Brazil, actually from the hospitals in Sao Paulo. So they actually include images like these, or rather, to make things a bit more concrete, let's go through the images themselves through the file. So they contain images like this thing right there. So this is basically a CT scan of lungs, and what happens is that we are able, or at least doctors are able, to point out whether a person is suffering from COVID-19 or not through such images. And one of the first things that we notice is that this one, as well as the other images, and the same pretty much goes for the healthy controls, because these images are taken from people that are healthy and do not suffer from COVID, is that they are grayscale. In other words, they do not really have colors, and they basically have, shades of gray pretty much in terms of their coloring status. And the reason why I mention this is because if we actually sit down and think of, images in a computational manner or in a machine learning manner, what we could say is that an image is nothing more than a matrix, essentially. It's nothing more than a table where its row and column represents a specific, color value, so to speak. So basically what we have is, again, we're working with tables. Images are tables for us, and the values that they include, which span from 0 all the way out to 255 for grayscale images, for gray images like the ones we've seen, represent different shades of gray. So 0 represents black, 255 represents white, and everything in the middle again is a shade of gray. If we extend this to like RGB or colored images, if you prefer, what happens is that you basically have a stack of different tables. So instead of having one table, imagine that my hand is a table, you basically have a stack of three tables, one right above the other. So pretty much like a sandwich of three tables, so to speak. And each table actually contains values for a different color channel. So you have one for red, you have one for green, and you have one for blue. And the reason why we have these three colors is because if we actually use these three colors and their different shades, we can create all other colors, at least algorithmically using, computers and whatnot. So this is really the difference between the main difference between a tabular data set in the sense as the one that we've seen with Iris or with the biomedical example from last week and what we will be working with today. Essentially, what I'm trying to say here is that although it might seem that images are an entirely different thing, at least when it comes to computationally speaking and programming, they really are not. We still treat them as matrices, as tables. So with that being said, this is a data set again from Kaggle, just to basically describe it a bit further. And you can always download the contents of the data set, meaning the images, by basically clicking on the download button here. So before doing this, however, you will have to sign into Kaggle, and you can do this by either using a GitHub account or you can use your Gmail account. I've actually used Google to do this myself. So with that being said, as you can probably see here, let me zoom in a bit. So I think this is going to be a bit better. We have 2,482 CT scans. We have these many images for COVID-19 patients and this many images for people that are healthy controls. So let's start seeing how we are going to process the data set and how we're going to move forward. So the strategy that we are going to employ here in terms of the way we're going to write the code is pretty much the same as what we've seen over the course of the past, lectures. So again, we have a Jupyter notebook. The first thing that we got to do is import every single necessary library, and you see that there are a lot of different tools that we use here. And you can see that we're also using TensorFlow, which is one of the two most well-established libraries for creating neural network architectures in Python, the other one being PyTorch. Then we basically start by initializing our links. Again, this is necessary because we have an external data set which consists of two different files for COVID and non-COVID images. We also create or initialize utility functions. So this is a specific function that we have created in order to be able to show our images or be able to display them so that we can actually check out what's going on within different pre-processing steps of the images that we will be using. And the first thing, aside from being able to initialize functions and libraries, of course, is to be able to read the data set. So we are actually using OS in order to be able to get into the file path that we have available where the images actually live within our disk. And you can see that the result of this code is this sort of initial data frame right there. So this is not what we will be using. This is just a catalog which shows us how images are going to be read in memory. And what can actually be seen here is that images actually have a specific order. So what I mean by that is that the first images that are seen or displayed in the catalog itself are COVID-19 images. So this is not really something that we would want to have. We would actually prefer to have like a shuffled image, at least in the head, in the first 5 values. And the reason behind this is because we want to have our images to be at random. So we would like to have images that belong to either COVID-19 patients or healthy controls. And the reason behind this actually stems from the fact that later on down the line, we are going to split our data set into training, test, and validation subsets. And we want to make sure that each of these subsets actually contains a fairly balanced amount of images that either belong to category COVID or to category non-COVID. So again, as a small parenthesis here, because we have two tags, we basically are working with a binary classification problem once again. But this time we're using images, of course. So to get around this issue that I mentioned right now, the first thing that we got to do is actually shuffle the way images are going to be read from memory so that it's going to be a bit more random. And if we do this by employing this piece of code right there, you can see that we have images that belong in both categories. And this is basically a random, initial distribution, at least for the first 5 images of our data set. The random seed that we use here also is a number for those that are wondering that ensures that even if I restart the notebook, the kernel, the result is actually going to be the same. So images are going to be read in the same manner. They're going to be random, so you're going to have images from both categories, but they're pretty much going to be the same. And the reason why we do this is in order to basically have repetition in our experiments, in case we want to hand them out to other collaborators, other researchers, so that they can actually be able to reproduce the results that we're getting. So after doing this, after being able to read our images and being able to shuffle them, the next thing that we got to do, again, is pretty much what we've done with other data sets, with the other tabular types of data sets that we've seen so far. So as you can see here, we once again employ a bar plot which showcases the number of samples or images in our case that belong into the two different classes that we have. Just as a reminder, class 0 right here is COVID, and class 1 is non-COVID. So what we can basically see here is that we have an almost equal number of images for both of our categories. And this is actually something very important, as we've already relayed in the past lectures, because this means that we have a fairly balanced data set, and thus we are not, or rather the chances of running into issues of training bias are actually far smaller than what would have happened in case we didn't. So with all that being said, we then move on basically. So we create another function. The way I've written this here is not right. I should have probably initialized it further up, but bear with me on that. And we use it. So this function basically, I'm not going to get into a lot of details, but it's based on a library called OpenCV. So aside from using TensorFlow in order to create my neural network and then train it and then evaluate it, I'm also using OpenCV, which is this CV2 right there, as you can see, which is a very powerful library or package when it comes to working with images, not only biomedical images, but images in general, at least in Python, that is. And we're going to use this function in order to be able to see or display our images. And what we can basically see here is that we've created sort of a table that includes 25 images. So the reason why it contains 25 images is because we have 5 rows and pretty much 5 columns. So it's basically like a 5x5 matrix that includes images. And this time you can see that it's from category COVID. So one of the first things, so rather, let's make it a bit more interactive. So aside from the biomedical context of the images, I mean, is there something that you would actually notice? And I'm not talking biomedically. I mean, I'm not a doctor or a biologist, so I wouldn't really be able to say why these people suffer from COVID. But when it comes to the images themselves, right? Is there some sort of difference between them? Something that we would actually notice that could perhaps be a potential problem, later on down the line. So what do you say? Would anyone like to try out? I'm also going to switch to the chat. So nothing so far. Okay, so the one thing that we actually see that's different, just to make things crystal clear, is that these images actually have a different size. Okay, so you can basically see here that this image, for example, is a bit wider, but you basically have a smaller height over there. This has a higher height and whatnot. So the problem with these images is that they do not share the same dimensions. And this can actually be a huge problem when working with neural networks. It is a huge problem with neural network architectures, as well as with any other machine learning algorithm, because the models or the tools that we use require a certain standardized input. So the fact that we have varying types of input or dimensions in our case is not really helpful, or is not going to be helpful at all for our neural network architecture. So we're actually going to have to find some sort of way to work around this. We're going to have to find some sort of way to basically resize the images that we have here at hand. And the same pretty much can be said in the case of non-COVID images. So once again, you can see that there are subtle differences, but they are differences nonetheless. I mean, we have different, sizes over there, shapes and sizes. So this can actually create, a huge part of confusion when it comes to working with such data. And as an example right there, you can also see what I'm actually relaying. So you have an image, for example, this one, which has a size of 370 by 326 by 3, and this one, which is 378 by 331 by 3. So these two values are width and height, and this one is actually the number of channels that we have. So what we see here is that we have images that actually include three different channels. And this might be some sort of point of confusion, right? So why do we actually have three channels? This would actually be something that we would see in like RGB, like colored images, right? With the sort of sandwich of matrices, each of which corresponds to a different color channel. But why would we actually do that over here? I mean, what we are working with is basically single colored channel images, because they are grayscale. Why would we actually have three channels? So I'm going to put a pin in this, and we are going to check out a bit later why we're doing this. It's more of a workaround for the model that we will be using, but I think that it's actually a very clever thing to do, and you'll see why in a bit. So having said that, because of the fact that we have different image sizes, once again, we have to be able to resize them all to a certain specific size, which is going to be standard, and it's also going to serve as the input dimensions for for the neural network architecture that we will be using. So we're basically, in a very high level manner, we're using this piece of code right there in order to ensure that we're going to read our images, and we're basically going to create them as 64 by 64, and we're basically going to change their sizes. And what's basically happening through all of this, just to um, basically kind of work around it together here without the code, is that if an image is actually larger than 64 by 64, it's actually going to be a bit compressed, like smaller. If an image is smaller than 64 by 64, if the dimensions are actually smaller, what's going to happen is that through the piece of code that we have right there, a padding is going to be created around it. And the padding is basically, essentially stacks of zeros that we are going to wrap our image around. So it basically is the equivalent of taking a small image and giving it some sort of background. In our case, it's going to be a black background. And the reason behind this is because, as you can probably see, we basically have the area of interest, which is within sort of a white envelope, which is the long area. And this pretty much applies for all of our images. And this is um, why we are choosing a black background in order to be able to reshape um, the image size, or to make it more standard, so to speak. So again, this code alone actually takes a bit of time. I've already run it, so the result of it is we basically get 2,481 images, the ones that we start out with, but now they're all at size 64 by 64. And again, we have 3 channels, but we are going to check out later on why we actually have these 3 channels. The other thing that we really need to do in our case, or rather, it's actually kind of optional because we already have zeros and ones, but it's an interesting thing or an interesting alternative to check out, is to be able to change our disease ID or the target variable in our case. So our target actually looks like this. It's basically a one-dimensional vector of zeros and ones, pretty much what we've seen before. But you can also use this function right there, which will basically turn it into sort of like a 2-dimensional vector. Okay, so you basically have values of ones and zeros. So you're basically one-hot encoding your target variable. So for example, imagine that there are 2 different columns for sample number 1. So this particular vector corresponds to the first image of our data set. And then you have 2 columns, one of which actually corresponds to category 0, and the other one corresponds to category 1 right there. So what this basically tells us, this 1 right there, is that if we imagine that there is a 0 up top, is that this first image actually belongs in category 0, and simultaneously that it does not belong in category 1. And it might seem a bit redundant to do something like this, at least in this particular case where we have a binary classification problem. But this also serves for, by extension, other kinds of classification problems, like multi-label classification, for example. So in that kind of situation where we would have multiple labels, you would actually be able to have multiple features that you need to check out or highlight within an image. So for example, it could be whether the person suffers from COVID or not, whether the person, for example, has lung cancer or not, and so on and so forth. So it's, again, in our particular case, because we were occupying ourselves with a pretty simple problem, which is, again, binary classification. It seems redundant, but we are actually doing it nonetheless so that we can have it as an alternative, or so that you can basically see that we can do this as well without any sort of issue. Um, I actually kind of went ahead of myself and I went on to a monologue. So, is there any particular question on in terms of the way we process our data, or perhaps in terms of why we are using label conversions, or anything at all that you'd like to discuss up until this point? So, um, is there any question, or rather, um, is everything understandable up until this point? This is the true challenge, or the true question. So I'm switching here to the chat so that I can also check out whether someone is writing or not. Okay, so I imagine that everything is that so far so good up until this point. So okay. So what we've actually managed right now is to get our data set. We've resized our images. So these are the initial images that we start out with. We resize them later on down here. We convert our label, of course. And then what we need to do is actually split our data set into train, validation, and test, subcategories or subgroups. The reason why we do this, once again, we are going to use the training set in order to be able to train our algorithm or model. We are going to use the validation part of the dataset or the subset in order to be able to validate or be able to test out the performance of our model during training. And then we're also going to use the test set, which is going to be a separate small subset with very few images that our model has never seen before during training or the internal validation. So we are using, again, this is pretty standard. This is what we've done before by using Iris or the other biomedical data set that we've worked with in our previous lecture. So it's pretty much the same kind of technique. And you can see here that we basically have the shapes for training, validation, and testing subsequently. And you can also check out here the resized images that we have right there. So even though they seem kind of pixelated and they do not seem as high quality or in as high resolution as the ones that you see up top, the nice thing that we see here is that they are actually of the same size. So they pretty much have standard dimensions, which means that we can use them as input for a specific neural network architecture. So there is a trade-off always when it comes to working with images as of course, with any other type of data set. So we might lose a bit of resolution quality in this case, but at the end of the day, it doesn't really matter because we want to have standard inputs no matter what. Okay, so this is the first part with the data that we have. So we've downloaded our data set, we've pre-processed it, we've displayed the images that we have, we resized them. So now we are actually going to move on with neural networks. So artificial neural networks to be more precise. So the basis of artificial neural networks is this thing right there. So this is actually called, as you can see here from the notes, the perceptron. And this is basically a mathematical function that serves as a model for neurons of the human brain. So the thing that Rosenblatt et al. That they actually tried to do is model the way our brain works. So the way neurons fire up and send signals to each other. So this is basically nothing more than a mathematical function, so to speak. So as you can see right there, you basically have inputs. In our case, imagine that the input would actually be some sort of vector. Again, we're not talking about images so far. We are talking about a classic neural network architecture. We are going to extend everything that we're going to say about neural networks, a bit further on for images. So imagine that an input, for example, in our case, is like a one-dimensional vector. And in each of these input green values that you see here, we feed a specific measurement for a particular feature of our vector. Each of these values is then multiplied by a specific weight. So what the weights actually do is that they determine the significance or the importance of its input value for this particular neuron in our model. We also add the bias term. The bias is actually an internal sort of fail-safe for the neuron so that we could actually have nonlinearity in the model. And this is important in order to be able to mimic very complex patterns of our neural network. We, of course, add all these weighted averages over there that you get, all these products of multiplication, as you can probably see right there. And then the entire result is actually fed through this line right there. This is actually what's called an activation function. And what happens is that this is basically the converter of our signal. So this is basically what determines later on whether this neuron is going to fire up or not and send its signal to other neurons in our network. And this is also a tool that we use, the step function in this particular example. There are other activation functions as well. But the reason why we use activation functions is, again, to be able to add an even further degree of nonlinearity to our model so that we are able to account for very complex patterns that are hidden within our data. So the whole promise of neural networks is that you're able to mimic the human brain, so you're able to train them so that they can actually infer and make predictions in very complex tasks where other machine learning models would not be able to perform all that well. Okay, and you can basically see here some of the nodes that we have right there for bias or activation functions. So a neural network, at the end of the day, is nothing more than basically an aggregation or a group of perceptrons. So this is a very simple neural network architecture. Neural networks are also called multi-layer perceptrons. And the reason behind this is because, again, the building block is the perceptron. So the only thing that we are doing is basically stacking them up in layers so that we can create very complex architectures. So, and depending on the architecture that we have or how many layers of these perceptrons we actually use, we have something like this. So we have the input layer. This is where the input or the values of our features and samples are going to be fed into the network. This is called the hidden layer because this is basically the intermediate part of our neural network, which includes, many different perceptrons, as you can see here, or neurons, whatever you want to call them, artificial neurons, that is. And then we have the output layer, which is basically what comes in contact with the outer world, so to speak. So the two parts of the network that come in contact with the outer world are, of course, the input layer from which it receives feedback and the output layer from which it actually gives out or fires up signals or feedback, or in our cases, predictions, right? So if we actually have many, many different hidden layers in this intermediate stage, so for example, if we had like 3 or 4 of these hidden layers, like one underneath the other, like a stack of them, then we would be calling this a deep neural network. So deep neural networks are nothing more than architectures that look like this, but with many more stacks of artificial neurons, one right underneath the other, which means that we have a very complex nonlinear model that is able to, at least theoretically at first, infer and make good quality predictions for very complex problems and data sets. And this is where the whole deep neural networks and deep learning, nomenclature actually comes from the architecture of such, models with the hidden layers, that is. So just to make things a bit clearer in terms of how these neural networks are actually trained. So what's the goal for training them? So let me um, actually check whether we have this right there because it does not play through the HTML. Okay, so how do you actually go on about to train a neural network? I'm not really going to delve into a lot of detail in terms of what's going on mathematically. We're going to check out how it's happening in a very high level manner. So imagine that you basically give a particular objective to the network itself. So the objective, which is this line right there, is called the loss function. So the loss function in a neural network basically showcases how bad of a prediction the neural network, or predictions if you prefer, the neural network actually performs, which means that if I train it and go on to use it in order to infer whether someone, in our particular case, actually suffers from COVID-19 or not, how wrong is our neural network going to be? So the goal when it comes to training such networks is to be able to minimize the value that we're going to be getting out of this loss function right there. And you can see that ideally, I would like the loss function, or the value that I receive at the end, to be all the way down here in 0. So a 0 error would actually indicate a 100% accuracy. But in real world scenarios, the truth is that 100% accuracy is not always, if ever, achievable. So in a binary classification problem, even though we are working with images, even though we are working with artificial neural networks, so in such a problem, an accuracy of like 98, 99%, perhaps if the problem is actually quite simple, is um, more than optimal. And 99 can be extremely optimistic in a lot of different cases. So the way this happens, the way we train our algorithm is, again, by what you're seeing here. So imagine that the neural network starts with an error function value up here. So you basically, how does it start? You basically start with a specific set of values for the weights of the network. So if we go back to the perceptron, we have a specific set of weights with particular values. The network, so what happens is that you receive input, we get the final prediction as we move forward right there. Then we basically evaluate the prediction according to a ground truth, and we get a value such as this one from our loss function. And what happens is that we go back to our network and we basically sort of say to it that, you know what, you're wrong. The thing that you've actually given us is wrong. It does not, um, contribute to the ground truth. It's not correct. So what happens is that we move backwards. So we move from the output, sorry, back into the input so that we can actually check out the weights that we've had in this first, part of our prediction. And what we basically try to do through all of this procedure, through all of this, is to be able to adjust the weights that we have down there so that we can actually get a lower value in our loss function right there. So this is called gradient descent and backpropagation. The reason why it's called backpropagation is because we start out again from the input and we move forward to the output, of course. And then to be able to adjust for the errors that take place in the output or the predictions that we have, we move backwards in order to be able to adjust our weights and get a better final prediction. So this whole procedure of going backwards and adjusting for these particular weights right there so that we can minimize the loss function value that we receive so that we can actually get into either a local or a global minima. In this particular case, if we speak in mathematical terms, is called backpropagation and gradient descent. So this is basically sort of the basis of how machine learning or deep learning models, in our case, artificial neural network architectures, are actually trained and by extension evaluated or tested upon real world data. I know that this is actually a bit confusing, perhaps. This is a bit of a hard concept, but bear with me. So now, having said all these, we were only talking about classical neural network architecture. So far we've actually seen a multi-layer perceptron. Okay, that's fine. Um, but why can't we actually use, or rather, the question is, can we use a classical architecture to work with images like the COVID-19 images that we have right here at the top? Like, is this possible? The answer is yes, but it's not really going to be optimal, like at all. And the reason behind this is because a neural network architecture such as this actually requires, in most cases, if not always, when we're talking about conventional neural networks here, that you have a flat input, sort of like one-dimensional vector representations of our data. But in the case of images, as you can see right there, we do not really have one-dimensional vectors. We basically have matrices. So flattening these into one-dimensional vectors is not going to be optimal. It would actually make for very, complex, very computationally expensive data sets, that is, which would actually require a lot, and I mean a lot of computing power to train and evaluate upon. And the second problem that these, more classical architectures have is the fact that they are not able to account for features within the images, at least when it comes to local neighborhoods. In other words, if there is a particular feature, for example, or a specific area of interest within this image right there, for example, this area that I'm showing my cursor on, a classic neural network architecture is most probably not going to be able to account for this. And this is where convolutional neural networks actually come into play. So convolution neural networks are basically an extension or an evolution of these networks, or artificial neural networks, and they're based upon this thing right there. So this is, again, a GIF. So this is actually called a convolution. So it's a mathematical operation. So what happens, as you can see, is that you have a sliding window, which is the yellow highlighted rectangle that you see here. And what happens is that we slide this small rectangle, this small filter, so to speak, on top of our image, which is this sort of binary matrix that you see here, the green one. And out of all of this procedure, what you get is a smaller matrix, which is this thing right there. So what happens through convolution is that you're able to use particular filters that live within your neural network architecture that are able to extract very specific features that have to within the image that are important for distinguishing very um, starting patterns, very simple patterns, that is. So this is basically the equivalent of being able to distinguish between, simple shapes and textures, like simple lines or shades within an image, at least when it comes to human vision, so to speak. The next layer that these actually have is ReLU layers. So again, we are using activation functions for this particular architecture. The reason why we use this particular function in most cases is in order to be able to grant a certain level, again, of nonlinearity. This is basically like saying, at least in terms of human vision, that I'm starting out by discriminating very simple, shapes and forms like dots, lines, what have you. And then I'm actually, trying to step up a level further and try to distinguish between more complex, non-linear features within my image, of course. And then there is also another class of layers that these networks include, which are pooling layers. And again, I'll show you a GIF right there. So a pooling layer, what it actually does is that it receives the convolved feature matrix. So you basically get this thing right there as an input to your pooling layers. Okay, going back there. And what happens is that you use another filter in order to be able to extract even more information and sort of like compress this feature map into a smaller one. So what happens is that this is the equivalent, at least in a high level manner, of making your image smaller the further you go into the network. And the reason why we want to do this is twofold. The first one is that we actually want to have a smaller image so that we can actually have fewer neurons, which means that we're going to make things a bit simpler computationally as we move on into the network. The second reason is because by resizing sort of the image, what happens is that we give the ability to our neural network to be able to distinguish between the important features within the image, no matter the resizing that we've created. Or in other words, we try to ensure that even though we might have, smaller images along the way, that our neural network will be able to distinguish between what differentiates between the images that we feed it with. And this is basically a full architecture, pretty much. Like this is an example right there. So what happens is that you have an image like this one right there. You basically have convolution. So convolution, again, is the ability to be able to extract upon simple features within your image. It's sort of like having a magnifying glass and being able to understand what is important for distinguishing particular parts of this image compared to other images, that is. You have max pooling, which again gives a certain level of nonlinearity so that we can actually extract even more complex features. And then you also have, or ReLU layers, that is, sorry. And then you have pooling, which can be done in many different ways. You can basically get the maximum value or the minimum one or the average one. This is why we have max pooling right there. This particular one receives the maximum value. And what this does is that this actually, as you can see here, once again, makes the image or the stack of feature maps that we have into smaller ones so that we can simplify our architectures and basically also be able to distinguish between important features no matter what they are, where they are in the image, no matter the size of the image itself. So, is that it? The answer to that is no. And the reason why it's actually not the case is because this whole part right there is basically the convolution side of our network. However, these layers that we've seen before, and you have many different, convolution and pooling and ReLU layers. So after stacking these one after the other, you then connect them to a standard, neural network architecture, which is pretty much something like this. So then you basically add this fully connected network, which will actually receive only the most important features from the images that we've actually extracted. So we basically perform a feature extraction, so to speak, with the previous layers. And then we supply these features that are important as one-dimensional flat vectors to the fully connected network, the standard architecture that you see here, so that we can actually perform classification. In our case, it's going to be binary classification, but you basically see a multi-class classification example from this picture right there. So it's basically like having the best of both worlds, so to speak. So you have convolution again, ReLU, and pooling layers, which are very useful when it comes to extracting, at first simple features from an image, then making them more complex and being able to distinguish or highlight them, whatever the size of the image or wherever they might be in the image itself. And you basically use all this extracted information as input for a more classical, second module, which will perform classification for you. So it's basically like having two models in one, so to speak. However, we basically call this CNN or convolutional neural network. And CNNs, just to make things a bit clearer perhaps, can be used for many different applications when it comes to image processing. So you can use them for classification, as you can see here. You can use them in a biomedical context, in other fields of research, such as image segmentation, which requires that you actually have a certain, set of images and that you actually highlight areas of interest within the image, areas like these ones right there, for example, and so on and so forth. So they are actually very well established within the research community, and we have many different architectures, many different shapes and forms, so to speak, of these networks that have been proposed, for different, types of work. And one such architecture, the one that we will be using in today's example, is this one. So this is basically a dense net. So in a very high level manner, what happens is that, as you can probably see here, in a CNN in a classical sense, each layer is connected to the very next one, and that's pretty much it. You basically have a sequence of layers where each is connected to the right next one to it. So what happens in DenseNet right there is that each layer, as you can see, is connected to the very next one, but it's also connected with deeper layers found later on or further down the network architecture. And it's basically like making sort of an ultra connection kind of situation where everyone is connected with pretty much everyone before them. And the reason why the idea behind this was actually done is that so that each layer can actually be able to learn, so to speak, and make use of pretty much the maximum amount of features that are extracted from previous layers. So in other words, the reason why this is done in the case of DenseNet, the idea behind it is that so that you can actually have a better final representation of your data in terms of images, as far as images go, so that we can actually get the maximum amount of information for our features. And this is supposed to make for a very robust architecture that can work with even the most complex of images. and even in the most complex of situations. Is this the only kind of architecture that does this? No, there are plenty of other ones out there. But for the sake of this example, we will be using this one. So how are we actually going to do this, right? And you can see some information right there. So this particular DenseNet that we're using has 120 convolution layers and 4 average pooling layers. Aside from the ones that we have, which are full of, simple perceptrons that will be doing the binary classification later on. So overall, we are talking about a very complex architecture that includes many different layers and actually has a lot of different parameters or many different artificial neuron weights. So whenever we refer to parameters, again, when we're talking about neural networks, we refer to the number of weights that we have that will have to be adjusted during training through the process of backpropagation and gradient descent. So what happens, as you can see here, is that we have a function like this one through which we're building our DenseNet. And what happens here, so this is sort of something really clever that we're doing. So we're actually using DenseNet, which is a function that is readily available through TensorFlow, and we're actually using weights that come from ImageNet. So what does that mean for us, right? What does this ImageNet mean actually? So when we actually go on and train a neural network, we can do this in two different alternative ways. So the first one is to build the architecture that we want to train and then use a particular data set from scratch in order to train it and then evaluate it. But the other thing that we could be doing is actually get a network architecture such as this that has already been trained in a large data set, or at least a data set much larger than the one that we have at hand, and then be able to use this already trained neural network architecture or model, if you prefer, in order to fine tune it or train it a bit further in our own, more specialized problem. So what we actually do in such a case, for example, imagine that this is sort of like our DenseNet architecture. And this particular DenseNet that we're using is actually trained on ImageNet. So ImageNet is a very large data set that includes colored images that depict very many different objects and animals and what have you. Back then, it consisted of about like 1 million images, something a bit more than that. So you can imagine a very, very big data set on which this particular architecture was trained. So what we actually do in our particular case is that we are getting, now again, imagine, let this be um, DenseNet 121, the one that we've actually gotten to use here. So what happens is that we have this pre-trained network where its weights, its convolution and max pooling weights have been adjusted on ImageNet. And what we're going to do with it is that we're going to sever it there. We are going to cut this multi-class classifier that we have at the end. And what we're going to do is that we're going to graft or stick our own binary classifier or our standard architecture to perform binary classification in our own set of images. And what we're basically going to do later on is train it on our much smaller data set that only consists of 2,400 images. So what we are doing essentially through transfer, and this process is called transfer learning or fine tuning if you prefer, is that we're getting a very big, complex network architecture that has been trained on huge amounts of data. In most cases, this is done by either very big universities or perhaps very large corporations like Google or Meta or what have you. So we are basically sort of exploiting the fact, that the big players are training very complex networks on extremely large data sets, and we are using this already existing knowledge so that we can adapt it to our own, set of images in this particular case. So our own smaller, perhaps simpler, or more restricted, problem, restricted in the sense that we have much less data to work with because we are working with about 2,000 images and the original DenseNet was trained on 1 million. So again, the reason why we're doing this is because we want to be able to use or exploit the already existing knowledge that exists out there for being able to distinguish between features or areas within images. And the second reason is also because of the fact that transfer learning and fine tuning by extension is actually computationally far less expensive than than actually running this kind of architecture on a very large data set. And truth be told, in most cases, like in our example with the COVID image classification, like with this data set right there, we might not even have that many images to begin with. Does that mean that we have to not train it and proceed? No. In this particular case, where our data set is limited or our computational resources are limited, what is actually much more effective to do is basically go on the internet, find an architecture that we like. There are specific repositories that actually include such models. They're actually called model zoos. And we basically get pre-trained models from these kinds of repositories and move on with transfer learning. We're fine-tuning these models to our own, particular problem. And this is what we're trying to do here. So we are building DenseNet 121. We are using the weights from ImageNet. So we have a pre-trained network. And what we are basically doing is that we are creating our own output or our own, let's say, head, so to speak. So this particular part is called the head because this is the final part of the network that will produce our output, which is going to be, of course, binary classification for COVID images, right? And then we also adjust, further parameters. For example, Adam is an optimizer that will help us, train the model or fine tune it in a more efficient manner. We actually have a specific loss function, which is categorical cross entropy. It's used. It's actually very well established for binary classification problems. And the metric that we use for evaluation, in our case, is going to be accuracy. So we initialize this function right there, and then we use it in order to build the model. Just as a parenthesis right there, this particular model can also be used or created through PyTorch, but we're, again, seeing how we can create this through TensorFlow. And right there, you basically have the number of parameters and a summary of the model. So you have 7 million parameters, which means that you have about 7 million 3,000 whatever weights, 300,000 weights. Trainable parameters is basically a reflection of how many of these weights we can actually train, how many of these weights we are able to fine tune. The non-trainable parameters are these ones right there. So you have about 86,000. And this actually represents weights that we cannot modify in any sort of way. In other words, these are weights that correspond to very specific, types of information that has to be extracted from an image. These are weights that have been trained upon ImageNet again, but we do not really want to change them because the piece of information that they carry or the kind of information that they will be able to extract through convolution and pooling and whatnot is very important, at least when it comes to distinguishing between our different images. And of course, before we move further with training, the other thing that we need to do is proceed with data augmentation. So what does this actually do? Let's move up again to our images. So we have images such as these. What data augmentation actually does is that it will create, geometric variations of these images. So imagine that you have these three images, for example. So what augmentation is going to do is that it's actually going to flip them either vertically or horizontally. It's going to rotate them to a specific number of degrees, either on the right or on the left side. And in some cases, it can even change the color space, for example, going from RGB to HSV or whatever. And the reason why we do this is because we want to supply our neural network with many different instances and variations of our existing images so that we can actually make a very robust final model that will not, confuse itself even if we actually supply it a slightly different image in terms of format. Like even if the image is slightly off by a few centimeters or rotated quite a bit. So, oh, and also something that I forgot to add is that the reason why we have images that are, three-channeled ones, like RGB images, so to speak, even though they're grayscale, so we have three channels for each of these, is again because of the DenseNet architecture that we are using. Because of the fact that this has been trained on ImageNet, which includes RGB images or colored images, which include three different channels. We could actually be doing one of two things. So we could be taking apart the entire architecture that has already been trained and basically change the input from being three-dimensional to being one-dimensional. But this would actually take a lot of time. It would actually be very hard to do. So the workaround, for this is that instead of, tampering with the architecture of the pre-trained model, we're actually adding two additional layers to each image so that we can have three-dimensional representations for each image. So we basically have three copies or three stacks of these images, one atop of each other. So we are basically giving our model sort of like pseudo RGB images to work around the fact that it has been trained or pre-trained, in our case, on ImageNet, which is an RGB dataset. Okay. And after we basically move on through all of this, after creating our generators for augmentation and whatnot, we can move on with model training. So we basically use this fit generator function in order to be able to use the augmentation part of our data as well. Just as a parenthesis again, right there, this fit generator function is now pretty much deprecated or old when it comes to Python code. There is actually a much simpler fit function, which does not include the initialization of our generator. However, the reason why I'm showing you this as is right now is because if you actually browse through a lot of different already existing projects, either on GitHub or Kaggle or what have you, um, at least when it comes to TensorFlow, they might be using legacy code such as this. So the reason why I'm showing you this is that so that you can actually get a very good idea of what code exists out there and not, confuse, yourselves in case you run into something like this. So model training is kind of boring in the sense that you basically get numbers for different epochs. So it's basically like what we've done before. We basically create repetitions of training and validation cycles so that we are able to train our model. And you can see pretty much like in the case of Iris before, in our classical models, you get a training accuracy, you get a validation loss, and then you get a validation accuracy right there. So the internal test set, so to speak, that was used during training achieved for our model like a 97.5% accuracy. And if we move on further down to the test set or to the images um, that our model has not seen at all during training or validation, we can basically see right there by making a prediction that it achieves like a 96.3% accuracy. So is that acceptable for our case? Well, considering that we have very limited, I'd say, images, it's a very good starting point. I mean, one of the very first things that we would actually do is supply the results, or rather, relay the results that we get into the medical professionals that we uh, basically collaborate with. And after that, we would go back and perhaps fine tune and change a few things into the network parameters, like learning rate, for example, or what have you, so that we can actually get potentially a bit better results than what we have here. And the final thing that you get out of all of this is, for example, like a confusion matrix between COVID and non-COVID samples. So the reason you see very few images right there is because this particular confusion matrix is based upon the test set of our data, which included like 248 images in total. So we can basically check out that we have a very high degree of true negatives as well as a very high degree of true positives. And we can also check out that we have a very small number of false positives and false negatives as well. So this seems to be kind of an ideal situation, but it should be approached with caution again, because even though our data set is balanced, even though we've been able to shuffle between our samples or images in this particular case before, splitting it into training, testing, and validation subsets, again, it's a fairly small data set, at least by today's standards. So in order to be able to move on further and check out and be able to have a very good concrete picture of what's going on for this particular model so that we can basically then deploy it into some sort of platform or a website or whatever, is that we would actually have to test it with even more samples. And of course, you can basically check here the curves that we get for training and validation. This is the accuracy curve. Um, you can see the training is a bit more stable compared to validation, which is a bit more violent, so to speak. The same goes for the model loss. So you basically have sort of like a stable, decrease in loss during training. This blue line right there, and this orange line is actually again a bit more violent, but we achieve a fairly low uh, loss score right down there at the end of our training sessions or epochs. And then, of course, after all of these evaluation plots and whatnot, we can read a specific particular image from our files, from the folders that we have, just as a final, sort of test. We do know that this particular image corresponds to a person that suffers from COVID-19. So we make a prediction and we also sort of normalize the image so that we can check it out. And what we basically see here is that the network architecture actually predicts that this image right there actually corresponds to a person that suffers from COVID-19, which is something that we would expect to see if the network has been trained correctly. So this is a giveaway that we are on a very good track, at least when it comes to training and evaluating our model, at least as a point of starting. So we would actually have to check further on um, what goes on with even more data, so to speak. And then, of course, you can see some citations or some references for further studying about what CNNs are and what TensorFlow is and how you can use this. So I'm going to stop sharing for a bit. So um, before we move on with the other example, I'd like to ask, is there anything in particular that you'd like to discuss? Are there any questions on this? Is everything understandable? Is there, I don't know, a particular point of confusion, perhaps something that you'd like to discuss, either through the chat or by opening your microphone?
It was understandable, I think.
Okay, okay, great. So let me also open up the second example that we have for today, which is going to be about electrocardiogram. Um, oh, okay. Thank you, Andrea. So once again, if you do have any sort of question, and this applies to every single one of you, feel free to interrupt me and open your microphone, and we can discuss pretty much anything you want. So um, without further ado, let me also share my screen to the second one. So let me make sure that this is the correct example. Okay, yeah, this is it. We got it. Okay, so I imagine that you can check my screen once again, right? Is that correct? Could anyone confirm?
Yes.
Okay, great. So this is actually going to be a simpler example. So the first one that we've seen has to do with biomedical images. Okay, this one has to do with biomedical signals, right? So the difference is that we are now working with electrocardiograms. And what is also quite a bit different in this particular case that we're going to check out now, in this use case of neural networks, is that this particular network, is not going to be used for classification or clustering or whatever. We're actually going to be using an artificial neural network in order to basically denoise a particular noisy signal. So the problem, and again, we're going to run through the code a bit later on, but the main problem with electrocardiograms is that they're actually very noisy when they come out of a particular machine that is being used to record them. So you have all sorts of different noises that are pretty much aggregated onto it. So you have baseline wonder noise, for example, which comes from the pulses of the heart. You have electrode motion artifact, like the electrodes that are used may not, stick up well onto the skin of the patient, for instance. You have muscle contractions. You have white noise that comes from the fact that the person might be stressed out and they might actually, move a bit during the examination. And you also have things like power line interference. So you basically have um, many different types. So power line interference, just as a parenthesis, is basically the noise that you receive from the electrical outlet to which the particular electrocardiogram recording machine is plugged into. So you have a lot of different types of noise, as you can see right there. This is a clean electrocardiogram, by the way, which, if added to the electrocardiogram, result into something like this. So you basically have a very, not chaotic, but a very nuanced kind of situation when it comes to working with electrocardiograms. So in previous years and over the past like 3 or 4 years, and even today, that is, the most classical way of dealing with these particular types of noise, at least when it comes to ECGs, would actually be to have some sort of different filters that correspond to different types of noise. So, for instance, for this particular baseline wonder, you would actually have a tool, a computational tool, that is, like an algorithm that would cut every single um, frequency that corresponds to this particular type of noise. Then you would have a second algorithm that would cut off the noise that comes in this particular band of frequencies for electrode motion artifacts. Then you would have for muscle contraction and so on and so forth. So you would end up with a very, complex tool that includes many, many different filters that are pretty much stacked on top of each other or right next to each other, whatever you want to call this, so that you're able to remove the amount of noise that exists within, that lives at least within the electrocardiogram that you have, so that you can actually supply it to doctors and they can actually make a very good, precise diagnosis of what's going on. So the idea of this particular project right there is, what if we can actually use a simple, neural network architecture, so that we can actually create an adaptive filter. So instead of using many different filters, each of which is going to be, sort of specialized by hard-coded values to a specific band of frequencies when it comes to filtering, what if we can actually use an architecture that can pretty much filter every type of noise at once? Right? So this is actually quite interesting. So again, we are using a Jupyter notebook. Okay, this is pretty much standard by now. The sort of strategy that we use is the same. So we import our libraries. This time we're importing WFDB. This is the database from which we are going to be getting our clean electrocardiograms as well as the noisy signals in order to add them to the cardiograms. Again, this is a project that is done in TensorFlow, but it could have been done in PyTorch as well, but whatever. We move on with initializing our utility function. So we have a normalization function like this one right there. And the reason why we do this is because in a lot of cases, normalizing your input, at least when it comes to one-dimensional samples or instances for classical neural network architectures, is something that is recommended as a very good practice. And we also have a denormalize function, which will help us receive the final part of the signal that we get, so that we can compare it with our ground truth and whatnot. And then we basically have this particular one, which is a sliding sample. So this is basically a sliding window. So what does this do? In this particular case, we are working with ECG signals, right? We're working with a one-dimensional sort of array. This is a signal that we have, which is in relation to time. Okay. The problem, however, is that the signal that we have, or the signals that we might be able to use, again, pretty much like the images that we used before in the CNN, might not have a very specific or fixed length in terms of dimensions. So what we're actually doing by using the sliding samples function is that we are actually trying to break down the signal that we have to a specified window size. And the reason why we do this is we basically do it to standardize the input that we will be giving to our neural network. So at the end of the day, whether we're working with classical tabular data, or rather in that particular instance, we wouldn't have such issues. But when we are working with less tidy types of data like signals, time series, that is, again, a signal is a time series, but whatever, images perhaps, and so on and so forth, what we are trying to do is standardize the input that we have, either by resizing in the case of images, for instance, or by using sliding windows in this particular case, of signals, that is. So the first thing after initializing our functions is to be able to load our database. Where this is basically the repository, the MIT BIH arrhythmia database that we use. This is where we're going to get our electrocardiogram from. And then we are also loading the noise database. This is actually a repository that includes different types of noise. For example, like baseline wonder, or electrode motion artifact, or muscle contractions. And then there are also different types of noise, like, for example, power line interference, which are not available in such repositories, but we can create it ourselves. We can basically check the bibliography in terms of the distribution of values for this particular type of noise. And we can basically use a specific kind of frequency like 60 hertz, as you can see there, which is pretty much the standard number that we use to represent power line interference. The same could be done for the other types of noise that we have, but since they're already available within a database, we simply extract them and use them on our signal. So again, as you can see here, we are pretty much initializing some parameters. We're also splitting a particular signal into training, test, and validation sets. And then these are the parameter initializations, and then we perform these steps right there. But let's not get all too technical in terms of the code that we have. These are the types of noise that we have right there. So the ones that we've talked before. This is the clean electrocardiogram that we have. This is one particular instance that we will be using for training. This is the part that is going to be used for training. So the first 18,000 seconds of it are going to be used to train our network. And then you can also see its noisy version. So what happens is that we are going to give this particular signal as input to our neural network. And then we're also going to give this particular one essentially as the output. So by giving it the noisy input as well as the clean output, what we would expect from our neural network is to be able to find a way to create some sort of a hybrid, dynamic, whatever you want to call it, filter through which it will be able to remove all different types of noise that exist within our data. Okay, so you basically see that we normalize our signals so that we can again feed them to our neural network. And then we create the train test splitting right there. So we split this particular signal right there that we have. Again, this is pretty much the same thing as we've seen before. So the clean signal as well as the noisy one, this is pretty much the same. We build a very simple neural network. So this particular architecture was actually proposed in a paper that was released in 2021. However, it was not implemented. It was simply, or rather, it was implemented, but it wasn't made available by the authors. If I recall correctly, it was actually done in MATLAB. So we wanted to test it out in Python. So this is what we're basically doing here with this particular architecture. And the reason why I'm relaying this is because in a lot of cases, just because you want to use a neural network does not mean that you have to think of the entire architecture right from scratch. In a lot of situations, you can start with something that has been proposed bibliographically, be it a simple architecture such as this, or something like a DenseNet, as we've seen before with the images. And after training and evaluating it and seeing whether this particular architecture fits or meets your needs or not, you can either keep it as is or modify it with certain elements in order to be able to make it more suitable for your own use case, that is. And then, of course, you basically see the same kind of deal that we've been doing with images. We initialize some parameters that are important for the backpropagation and gradient descent steps so that we can actually train our model effectively. And then we proceed with training, which again will give us some certain results when it comes to loss and mean squared error and whatnot. You're also going to check out here the mean squared error curves through training and validation. But what is actually the most interesting part, at least for me, is this thing right there. So the blue signal that you see in the background is the noisy ECG. So the one that we have, the real one, after we've added all sorts of different types of noise to it. And this orange one is the cleaned out electrocardiogram as we've received it from our neural network architecture right after training it. So it might not be all that meaningful when we overlay it on the noisy signal, but what happens if we actually overlay it on the original signal right there, which is this blue one that you can see behind it? So what we're actually checking out is that although they are not exactly 100% the same, they are very, very, very close to each other. So this is basically an indication that although it would require some fine-tuning, our model, our particular neural network, has actually been able to account for at least as much of the noise as possible and remove it from the signal so that we can have a clearer picture of our final, data of our electrocardiogram. And what happens later on as a second use case for our particular study right there is that we remove muscle contractions. So this is something that was done because we already knew that for this particular signal, the patient was very calm and they didn't really have that sort of issue when the recording was happening. So we removed it completely and we check what's going to happen with the other types of noise that we have in the data set. And you can see that this is the noisy sample again, as we've seen before, but this time without the electrode motion artifact and whatnot. Again, this is the clean signal and then the noisy one. And if we move further down the line after training and inferring and whatnot, again, we receive a picture which is even better than the one that we've actually had before with the electrode motion artifact. So what we see here is that if we remove this particular type of noise, because we knew that this was not an issue during the examination for this particular patient, we're actually getting a very good representation of the real ground truth of our signal, which is pretty much the orange overlay that you see here on top of the blue one. So what happens is that instead of using, and this is actually quite interesting because it's a giveaway that just because neural networks have been created to be used mostly for classification or simple regression tasks or whatever, you can also use them in other fields of research, for example, like signal denoising, which might not be as obvious from, at least when it comes to first glance, that is. So I'm actually kind of getting ahead of myself. I'm talking quite a lot. So this is basically what I've had for today, these two particular use cases. So is there any question for this second use case? Perhaps something that you'd like to discuss for this particular architecture, the way it was trained, maybe, or rather something about signals, anything at all. Like, I didn't really get too technical with it because I've been rambling about neural networks all this time and I didn't want to tire you all that much. So, yeah, is there anything you'd like to discuss? Or rather, is everything understandable? You can give me any sort of feedback you want, either through the microphone or the chat.
Yeah, it was great. Thank you.
Okay. Okay, so that. Oh yeah, Andrea. Yeah, of course. Go ahead.
Thank you very much. I would like to only be sure that based the second method only on the images. I mean, was there any another symptoms? I mean, was the patient stressed, or was there any illness, or based on only on the EKGs?
Oh, so you're asking if I got this correctly for the ECG network that we used, right?
Yes, I mean,
If
was there any another additional information about the uh, people? I mean, illness or stress factor,
oh,
or
okay.
during the
Yeah, there were actually other types of metadata in the data set. Like this particular data set has to do with arrhythmia. So some of the patients are healthy controls, at least the signals that we get, and some actually do suffer from arrhythmia. And in these particular cases, you would actually get a slightly different signal. So you do have, different types of metadata. So yes, this is the answer for that. However, we decided that aside from the metadata, which is something that a lot of people might focus on, like, classifying particular signals and whatnot, what would be interesting, again, is to be able to use a network for pre-processing purposes. So they do exist. I mean, at the end of the day, you would be able perhaps to combine two different networks, one which would be able to account for the different types of noise and remove them from the signal, and perhaps a second one to make some sort of prediction for whether the um, particular signal has to do with stress or if it's just, basic or rather, severe arrhythmia and whatnot. But we actually focused mostly on the pre-processing part in the sense that we wanted to see whether a particular, fairly simple established architecture can effectively be used for, denoising. Because back then, this was actually a project back from 2021, I think. Yeah, 21. So back then, the king, so to speak, when it comes to denoising, where these, and they still are in some situations, some very complex computational methods that require many different field filters and many different frequencies to be used so that you can actually remove different types of noise. And the idea was, again, that instead of using this kind of, complex, messy program that includes many different parameters, why not just use, a fairly simple architecture, a neural network architecture, and pretty much be done with it? Like, will that, first of all, the first question was, will that work? And At first, we didn't expect it to work all that well, but yeah, here we are. But yeah, they again kind of a long answer like in previous lessons, but the answer is that there are metadata, and yeah, you could definitely use them for something. But again, this is basically dependent on the narrative that you have. It actually depends on what you want to make predictions on. So yeah, I'm sorry. It was a bit of a long answer. I hope it answers for you.
I like the long answers. Thank you very much. Thank you very much for the great overview again.
Okay. Okay. Thank you very much, actually. So, are there any other questions? I actually saw that someone was writing in chat, but I haven't really seen anything specific. So, whoever actually was writing in the chat, would you like to open your microphone and basically, say what you had in your mind, just as a part of the conversation? I mean, was there anything, or was it just me? I don't know. Okay. So I'm going to ask once again, is there anything else you'd like to discuss? Anything in particular, either about the neural network architectures that we've seen today, or perhaps for the course materials that we've actually checked, from the start of these lectures that we've had together? Is everything understandable? Is everything okay? I mean, in terms of the materials? I'm guessing that it is, since I'm not receiving any sort of feedback. So that pretty much wraps it up.
Yeah, yes. I think everything is all right.
Okay,
Thank
great.
you very much.
Okay, I'm really glad for it. I'm really glad. Thank you for the confirmation, Andrea. So with all that being said, this pretty much wraps it up for this particular seminar. Thank you all very much for being here with me and paying attention to what I've been saying. Sorry if I was actually a bit tiring at moments, especially with my long answers and analysis. So I hope you found things interesting, not only today, but over the entire course. And I wish that you all have a great Christmas, since Christmas is around the corner. So have a good holiday. And once again, if any one of you wants to reach out and make contact for a project or what have you, feel free to do so. You already have my email, you have my GitHub repo, so I guess we'll be in touch. So we are going to have a very short break, and then you're actually going to continue with the lecture with another instructor, another lecturer. So again, thank you very much for your attention, and I'll see you around. So have a good one.
Thank you. Happy holiday for you too.